Hi, everyone. Again, this is Janine Holly, and I think we're going to go ahead and get started. I am the manager of the NGS Leaders online community. We're pleased to bring you today's webinar where we're going to discuss a hybrid error correction algorithm that was the subject of a recent paper. Two of its authors will give us an overview of their work and answer your questions. Use the chat box on your screen to submit questions during the presentation, and we're going to try to get to as many of them as possible. Also, toward the end of the session, we will post a link to a short feedback survey, and would be grateful if you'd spend a few minutes to give us your input and help us to plan future events. We will get started in just a minute, but I wanted to give you a brief background on NGS Leaders for those of you who may be joining us for the first time. NGS Leaders is an online community that acts as a platform for networking and learning for people working in genomics and next-generation sequencing. In addition to webinars, some of our other features are discussion forums, a blog, articles, and a poster gallery. So if you haven't done so already, please spend some time and take a look around the website. NGS Leaders is managed by Cambridge Health Tech Associates, or CHA, which offers consulting and technology evaluations along with our online communities to help clients in various areas of the life science industry to achieve their individual goals. If you're interested in learning more about CHA, you can let us know through the survey at the end of the webinar. And I'd also like to mention two conferences that may be of interest to our audience today. They are produced by our sister company, Cambridge Health Tech Institute. There's a Next Generation Sequencing Conference next week, in fact, in Providence. And there's a Consumer Genetics Conference, and that's in October in Boston. For the conference next week, we are happy to offer NGS Leaders members a last-minute $200 discount on registration if you enter the key code NGS200 when registering. So with that, let's get started. I'd like to introduce Kevin Davis, who will be our host today. Kevin is the editor of BioIT World and also author of several books, including, most recently, The $1,000 Genome. Kevin? Kevin, are you
Thank you very much, Janine, and good morning, good afternoon. Welcome, everybody, to this NGS Leaders Web Symposium. We have a great topic today with two superb speakers who have published one of the landmark papers in next-gen sequencing in 2012, and I will give you their background and hand over the reins to them in just a couple of minutes. The title of today's symposium is Hybrid Error Correction and De Novo Assembly of Single Molecule Sequencing Reads. As Janine mentioned, I'm Kevin Davis, author of The $1,000 Genome and the editor of BioIT World, which is a sister company of NGS Leaders, both of us owned and operated by Cambridge Health Tech Institute. So as I was saying, it's been an exciting 6 months in the next generation sequencing space. We've had a fascinating high-profile teasing presentation from Oxford Nanopore earlier in the year. We've just recently heard about the first official entrant in the X Prize $10 million competition. That'll be Iron Torrent. The competition will be held in September 2013. Another high-profile publication just in the last few weeks on long fragment reads from Complete Genomics. And as you'll hear in the next 55 minutes or so, a very powerful method for de novo assembly of not only small but also large genomes. Single molecule sequencing with long DNA read lengths, made most notably from the platform produced by Pacific Biosciences, has enormous potential for de novo genome assembly, but the rather high error rates associated with current technologies have presented some serious limitations in that regard. But a recent paper published in Nature Biotechnology described a correction algorithm that has been developed to address that problem. The authors were able to marry the long but slightly error-prone single molecule reads from Pacific Biosciences with the highly abundant and relatively high quality but short read lengths from the Illumina HiSeq instrument. This paper was co-authored by researchers from Cold Spring Harbor Laboratory, led by Michael Schatz, and NBACC, the National Biodefense Analysis and Countermeasures Center in Frederick, Maryland, and we'll be hearing from them in just a minute. As I say, they've been able to demonstrate the ability to assemble not only microbial, but also eukaryotic genomes with extremely high accuracy by combining the strengths of those two complementary platforms. So today we're delighted to welcome to this NGS Leaders webinar the two corresponding authors of that Nature Biotechnology paper, Adam Philippi and Sergey Koren, who will present an overview of their hybrid mapping strategy, highlighting several of its potential applications, as well as perhaps discussing some of the remaining challenges and areas for improvement. Just a brief introduction to our two speakers, and then I'll hand it over to them. But a reminder to all of you on the call that we very much want to entertain your questions at the end of the formal presentations. And to do that, you can enter your questions at any time during the next two presentations by simply entering your questions in the chat box that you see on your computer screen. Please do that at any time during their talks, and I will take all of the cogent questions at the end during the next 50 minutes or so. So our two speakers are Adam Philippi and Sergey Koren, both with NBACC, the National Biodefense Analysis and Countermeasures Center, as I say, in Frederick, Maryland. Adam is principal investigator in bioinformatics, which means he's essentially he is the leader of the computational genomics group. Adam received his PhD in computer science from the University of Maryland under the direction of Steven Salzberg, and has previously worked at Tiger, the Institute for Genomic Research, the J. Craig Venter Institute, and IGS, the University of Maryland Institute for Genome Sciences. Many of you may know Adam. He's developed a number of important bioinformatics tools, such as the MUMMA genome alignment software and the AMOS genome assembly toolkit. His research interests focus on microbial diagnostics and forensics using next-gen sequencing. Adam's colleague Sergey Koren is a bioinformatics scientist in the genomics group at NBACC. Sergey was previously at JCVI, the J. Craig Venter Institute as well, and recently received his PhD in computer science from the University of Maryland. His work focuses on genome assembly, metagenomics, and applications of high-performance computing to bioinformatics, and he is also a primary contributor to the Celera Assembler, AMOS, and MetAMOS projects. So with that, I'm going to hand the microphone over to Adam, and Adam is going to take things from here. Adam, thank you very much for joining us, and a warm welcome to you.
Thank you very much for that kind introduction, Kevin. We're glad to be here. As Kevin mentioned, we're here to discuss our recent publication in Nature Biotechnology. We're going to aim for doing a formal set of slides for maybe about 30 minutes, and then we're most excited to take any questions and answer anything that might be on your mind in the remaining 30 minutes of the presentation. So I'll start with an introduction to the problem, and then about halfway through I'll hand over to Sergey, who will present some of the results that were both in the publication and some of our most recent results with some of the latest PacBio chemistries. So first, Sergey and I are both currently located at the NBAC, the National Biodefense Analysis and Countermeasures Center. It's a relatively new institute. We've only been operational for about 2 years. Our primary designation is the lead center in response to any domestic bioterrorism event, such as an Amerithrax incident or so on. Sergey and myself are primarily concerned with microbial forensics. That's the forensic investigation of microbial materials, metagenomic materials, and so on in support of the U.S. government. Our odd-looking logo here is a depiction of our building, which is shaped like a ship for reasons unknown to me. Because we are a federal agency, we're an FFRDC funded entirely by the Department of Homeland Security, and as such, we are not allowed to endorse any products, so anything that we say in the following presentation come with no warranty or endorsements attached to it. So first, a very high-level introduction about our method and what our inspiration was. So I've included a painting here called The Alchemist as kind of a summary of what we're trying to do, in that we're combining two ordinary elements to get something that's extraordinary. So second-gen sequencing has been the landmark technology for the last 7 years, and the focus has very much been on short, high-accuracy reads that are very plentiful and huge throughputs. So this has driven down costs, increased our ability to do SNP genotyping and so on, But there are drawbacks in that the reads are short, so from transitioning from Sanger to second-gen sequencing, our ability to assemble these genomes was diminished quite a bit. In addition, because all of the second-gen technologies are based on amplification processes, there can be significant biases in both the error and the coverage observed. So that's our first basic element. The second basic element is the newly emerging third-generation machines, in particular the PacBio instrument, which has been available since last spring. And they're quite complementary to the second-gen technologies in that they're long and unbiased, but they have considerably lower accuracy. In most cases, a single pass read from PacBio has about an 85% average accuracy. And also they have low throughput. So our question was, can we play alchemists here and combine these two basic elements to get something that's the gold standard? So the goal being both long and accurate so that we can use the accuracy of the short reads to provide high quality base calls, but use the length from the single molecule sequencing to improve genome assemblies both by their length and their low bias. So really the complementary aspects here are what intrigued us in using this hybrid error strategy, and we set out to prove that the third generation error rates could be effectively managed by combining it with short second generation reads. And the results have been quite spectacular, so Sergey will present in the results our assembly of the parrot genome that produced an assembly that was about twice as good in terms of contig continuity as previous bird genomes done with Sanger sequencing even. So why does read length matter, and why do we want long reads? So I'm displaying here, as I bring up the laser pointer, assembly graphs from E. coli K12 genomes. So this is a very simple genome relative to some of the other complexities that are out there. And we've displayed 3 assembly graphs. So these are De Bruijn graphs, assembly graphs, string graphs, whatever you'd like to call it, constructed with different word sizes to kind of represent a range of sequencing lengths. So on the left, the smallest, k equals 50, would be a representative assembly graph if you had perfect, say, Illumina data of length 50 bases. In the middle here is k equals 1,000, so maybe a representative assembly graph for Sanger data. And to the right, k equals 5,000. This would be a representative assembly graph if we had perfect PacBio-sized reads. And this is from a paper of a previous colleague of mine, Carl Kingsford and company, that was published in 2010. And they were primarily concerned with assembly complexity of Illumina and other short read assemblies. As you can see on the left here, when you have read sizes of about 50 to 100, the assembly graph of even the most simple of genomes, like a K12, is extremely complex and filled with tangles. So each one of these edges that you see, the individual nodes you can think of as contigs, and the individual edges are relationships between, say, 2 adjacent contigs. In reality, all of those dots are contigs, and each of the edges represents potentially missing sequence information. So either missing in terms of you don't know which contigs go next to each other, or missing in terms of the assembler wasn't able to reconstruct a large enough piece of the genome, so you're left with a gap in your assembly. As you increase the read length, though, the assembly graph simplifies itself. So as the reads get longer and longer, the repeats that you're trying to detangle become less and less. So by the time you get up to K5000, there are no repeats in the E. coli K12 genome that are larger than that length, and so your assembly through a typical overlap-based process comes out as a single contig. So all of the assemblers of the past decade have been well aware that read length is king in genome assembly. It doesn't matter how much depth of coverage you have with Illumina data, you can sequence to 1,000, 2,000, 5,000x, and you'll never put the genome together if the repeats that are in your genome are larger than your average read length. So there's very important things to be had on these edges. So the smaller read lengths, you're not assembling everything. Each of these edges represents something that's missing. Typically that's intergenic sequence, introns, repetitive sequence, tandem repeats, all kind of structural, regions of structural importance in the genome. Generally the exons and the coding sequence are relatively unique, and they'll come out in full contigs. But it's kind of the structural content that we're most interested in using these big reads for. So the layman's analogy is if you're doing a puzzle, you want big pieces in your puzzle. So if you're trying to put together a Dora the Explorer puzzle with 10 pieces, that's much easier than putting together a Where's Waldo puzzle with 10,000 pieces. So the larger the pieces are, the easier we can do an assembly. So the question is, do we do that with the traditional overlap-based approaches, or do we do that with the De Bruijn approach, which has been the fad for the past 5 years? So our initial intuition was to go with the overlap-based approach because if we're going back to the puzzle analogy again, the overlap-based approach takes the puzzle pieces as they come into the assembler. So if you have a 5,000 base read, it gets treated as 5,000 bases. In a De Bruijn assembler, on the other hand, they generally shred the reads down to a fixed Kamer length that's shorter than the read length. So say the De Bruijn length, your Kamer size is 100 bases. Your reads coming in as 5,000 bases will get shredded down into 100 base pieces. This didn't make a lot of sense to us. We wanted to take the most advantage of those long reads, so we set out to go with an overlap-based approach. The keys being that the repeat complexity in your graph depends solely on the read length, not your choice of a Kamer. And also overlap approaches tend to be robust to error, whereas De Bruijn has some of the opposite aspects, where it's good at high coverage data, but in contrast, the repeat complexity depends on the word length that you choose. So if you choose a small k, you get a highly tangled graph. So on the right, this is a paper from our figure that shows a comparison of the Celera assembler versus SOAP de Novo for increasing read length, and this kind of illustrates the shredding properties of a De Bruijn assembler. So on the bottom, we're showing a PacBio simulated read length, where this is a perfectly accurate read that we've simulated at various lengths. And then we're showing the N50 contig size here in terms of kilobase pairs for the yeast genome. The black line shows Celera assemblers contig size almost linearly increasing as the PacBio read lengths are improved. However, the SOAP de Novo assemblies are flat based on whatever your choice of k was. So here's a k equals 63 and a k equals 127, which are the largest Kamer sizes currently available, and you see no improvement in read length. Now, this is somewhat of an unfair comparison because there was no modifications made to SOAP de Novo to take advantage of the long reads. However, there are some De Bruijn assemblers available that employ read threading techniques, which try to take your De Bruijn graph and detangle it using larger, more continuous reads. A lot of them are very computationally intensive. So for a large genome like yeast or bird or so on, it can be very difficult, if not impossible, to do a full read threading for very long reads. So the easiest approach for us was to use the Celera assembler, which already existed and was an overlap-based assembler, so that if we could correct these PacBio reads, we were confident that we would get very high continuity assemblies out the other end. I guess I'll also make this point to note. I'll bring up that All Paths LG had a paper released about 2 weeks ago that also showed support for PacBio data. In the next slide, I'll show our approach, but I'll just give a preface to the All Paths approach, since I'm sure many people have seen that paper. It does the opposite approach that we do. So we do error correction, and then we do overlap-based assembly. All Paths LG does De Bruijn assembly first on short read data, and then it tries to do this kind of read threading problem with the PacBio reads in the scaffolding phases. So we can talk more about the comparisons of those two methods in the discussion following the slides. So a brief introduction to our correction and assembly method. So our original intuition was if you start with two PacBio reads that have high error, it can be very difficult to find an overlap between the two of them. So if each of these reads has an error rate of 15%, the number of differences between any two of those reads will be 30%. It's almost an additive property. If you have 15% error in this, they don't line up with the 15% error in this one, and you get additive, nearly 30% error between the both of them. So in alignment terms, that's very difficult to do. You require a very high sensitivity aligner to detect that similarity, and the runtime would be very high. In contrast, we realized that aligning a very highly accurate Illumina read to a PacBio read would be much easier because the Illumina reads or the 454 reads are nearly 99, 98% identical to the truth. So if you're aligning one of those to a PacBio read, you only have to find about 15% differences, and that's within the tolerance that our aligners are able to handle. So the first step is to map all of those short reads to all of the long reads. Here, each of the blue boxes represents a short read. The red reads represent a PacBio read. And then each of the little tick marks represents an error in the mapping. So this is if you're running, say, a bow tie aligner, this is the number of errors that you would be tolerating. Our overlapping step uses the overlap code that's in the Celera assembler, which is a traditional seed and extend algorithm. Once we have all of these alignments of the short reads to the PacBio reads, we can look at the depth of coverage, so how many short reads are aligning to each spot on a PacBio read. And in cases where that depth looks unusually high, we have some statistics in there to determine whether or not we're looking at a unique region of the genome or a repetitive region. And we try and make a decision there to separate out any repetitive copies. So if I have a 2-copy repeat here, RNR prime, I have twice the depth of coverage in each of these reads on that repeat, because the reads from this one also map to the repeat over here, and the reads from this one also map to the repeat over here. So we sort these alignments based on identity. So we look for the short reads that best map to the long read, and then we determine a cutoff based on the coverage, and throw away all of the low identity, high coverage reads that are likely coming from repetitive copies. And this is in an attempt to get a correct reconstruction of not just the unique sequence in the genome, but a correct representation of each of the individual repeat copies. There might be subtle polymorphisms within the repeats that we want to make sure we get. Once we have that, we compute a layout. And importantly, we trim the coverage. We trim the original PacBio reads anywhere where there's an odd coverage gap. This means where there's a position in the PacBio read that has no Illumina reads overlapping it. This is an important step for us because the PacBio chemistry uses hairpin adapters on the end of its molecules that it puts into the individual sequencing wells. And the polymerase can read around that molecule multiple times, and this results in you having what looks like a chimeric read because you're reading the forward strand, you go around the hairpin, and then you're reading the reverse strand. If that doesn't get properly trimmed by the PacBio analysis software, that could confuse an assembly program because it will look like a chimeric read. The insight there is that we will have no short reads expected to map to those hairpin adapters, and we'll have no short reads expected to map across that chimeric junction. So anywhere where we see a region of 0 coverage in the PacBio read, we tend to trim that off because it's a suspected chimer. Once we have the trimmed reads, we take the second generation, either 454 Illumina or even PacBio CCS reads, and we compute a consensus sequence for each of the PacBio reads. So all of the PacBio reads that go in have a little mini assembly constructed for each of them. A consensus is called for every individual PacBio read, and then the output of that correction is output as a collection of highly accurate and corrected PacBio reads. These corrected reads then can be used for anything a normal read can use for, and this is kind of a key difference of our approach. Because we're able to do correction, we're not relying entirely on the Celera assembler. You could take the corrected reads and use them with any tool that you like. We've had good success using the Mira assembler, for instance, which now supports long reads, but it only supports high accurate long reads. So if you correct the PacBio reads using our approach, you can import them into Mira and it will assemble well. Also, traditional alignment programs such as the Mummer alignment package that I've developed over the years is very effective at aligning these high accurate long reads. You can use it for RNA Seek programs. And this is the key to our method, in that the PacBio sequencing technology itself has been heavily limited by a lack of tools that are available to analyze the data. What we've done is created a pre-filter step that essentially takes the high error PacBio data, turns it into high accuracy data that then can be used and analyzed with any of the many bioinformatics tools that were developed over the past 10 years. Typically, what we'll show in our results is after the correction, we're feeding those corrected reads into the Celera assembler, which uses an overlap, layout, and consensus assembly strategy, and we'll be reporting on the results of that assembly process. The output of these reads in our experiments is always about 99.9% accurate or greater. And the coverage that you miss, the length of the reads is not shortened by a terrible deal. But if there are a number of very inaccurate reads, they could be excluded by the pipeline. So maybe not all of your reads will get corrected by the pipeline, but the ones that are corrected and output will be very highly accurate. So a question that we're commonly asked is how much coverage is required to take advantage of this process. So here I've shown it from 2 angles: the required Illumina coverage, which represents the coverage you need in the correction technology, and the PacBio coverage required, which represents the coverage needed for the single molecule sequencing. The preface stating that you don't need to use Illumina for correction. Any of the second generation or any highly accurate short read technology is sufficient for correction. For instance, you can use 454 correction, which works quite well. You can use the PacBio CCS, which is their circular consensus sequencing technology, to correct their single pass reads. That works quite well, and Sergey will have some results on that coming up. So the key highlight for Illumina is that up to 100x coverage improves both the throughput, meaning the number of reads that are corrected, and the resulting assembly continuity, which is shown here in the percentage of bases that are corrected and the resulting contig N50 of an assembly as Illumina coverage increases. So based on this chart, we generally recommend between 50 to 100x of Illumina coverage for correction, and those results show diminishing returns as you get higher than that. And we don't recommend certainly anything over about 200x of Illumina. That'll just increase the runtime of the program unnecessarily. If you're correcting with 454 or PacBio CCS, 20 to 40x of coverage is sufficient. On PacBio coverage, the key result that we had from here showed that you get very rapid gains up to about 10x of PacBio coverage, and then after that you get diminishing returns. 10x seems like a very low number, but if you recall back to the age of Sanger, all of the whole genome sequencing projects, including mouse and human and so on, were around that number or even less. And so we're kind of returning back to Sanger coverage with the introduction of these long reads, where you don't need 100x of Illumina to get a high quality assembly. Just the addition of 10x, and in the case of Parrot, we only added 4x of PacBio reads, and we saw significant gains. So in the plot I'm showing here for an E. coli, this is one of the German E. coli outbreak strains, JM211. The introduction of just 10x of PacBio coverage took the N50 contig size from 100 kbp to nearly 400 kbp. So you're seeing a 4x improvement in the continuity of contigs with the introduction of just 10x of PacBio. I'll also note that this coverage shows diminishing returns, but if you want to get the very long repeats, say the rRNA operon in bacteria or large interspersed repeats in the human genome, if you sequence out to a very high depth of PacBio coverage, you start to get very long reads that can help span some of these. So you start to see a profile where you get very rapid gains up to 10x, it levels off for a long time, and then once you hit kind of a critical threshold of PacBio coverage where you start to get enough long reads, you'll jump over some of those repeats. And Sergey will have some results on that to present on the next slide. So with that, I'll hand it over to Sergey, and he'll talk about some of the results that we've had recently in assembling both bacterial and eukaryotic genomes. So if Janine doesn't mind passing the ball to Serge, it's all his.
Okay, thanks, Adam. So like Adam mentioned, I'm going to go through some of the results we have both from our paper and some more recent results. The first part, I'm going to focus on the bacterial results, and then I'll go on and talk about eukaryotic assemblies and what kind of improvements we can see in those using the PacBio long reads. So our goal for the bacterial genomes is to do automated closure. Typically closing a bacterial genome, even relatively simple ones like an E. coli K12, is very manual, very slow, and very expensive. So we want to address that and do it in an automatic and repeatable fashion. Our goal is basically to do this without having to generate any kind of long-range jumping pair libraries, so either Illumina jump libraries that are in the 3,000 to 10,000 base range, or 454 pair libraries which are in the 2,000 plus range, because these are typically very expensive and difficult to construct. Our secondary goal is to be generic enough that we don't have to correct the PacBio reads with one specific technology, so we don't have to have Illumina data or 454 data. Whatever secondary data you have, as long as it's high accuracy, we can use it and utilize it to close genomes. So that's our overarching goal for the bacterial genome, and I'll go through some results that shows our success with this approach. So the first slide here just shows some preliminary results that were done by PacBio themselves using their data. And the first three lines show an E. coli K12 genome. So this shows first the same kind of story that Adam showed before, that adding 14x of PacBio triples your N50 and doubles your maximum contig length, so just like what we were seeing in the previous slides. And they're close, no matter whether you correct with a CCS or with a MySeq data, the results are pretty close. You still get a significant improvement over MySeq data alone. However, in this case, the CCS seems to work a little bit better, though it's more expensive. So our goal is to find the sweet spot and say, if we want to use CCS data, does it work better, but the MySeq is significantly cheaper? So can we use cheaper MySeq sequencing? Do we need CCS data? And we want to explore those options and see which ones work best for assembly. So we had one genome which was a collaboration with the USDA, and this was the first closed genome that we're aware of that was automatically closed. So this genome is 2.5 megabase genome, and the largest repeats it has are the RNA operons. So these are in the 5,500 kb max repeat range. In order to assemble these with something like Illumina or 454, you have to have a lot of large libraries to span over these repeats. So you need to have something like a 7K or 8K library in the 454 data, and you need to have other libraries complementary with that to figure out the actual structure within the repeat. And this genome, the reason that we looked into this genome was because this was one of the genomes that they were having the most trouble with. So they were doing a large-scale study with several different strains, and they had the most issue with this assembly. And you can see that even when they did 454 8K library, it didn't really assemble well, even with that data. So what we did is we used our correction pipeline. And what we did is we basically just took the tail of the distribution. So the PacBio sequencer produces an exponential distribution of data. And we were interested in only the long reads, because if we want to assemble these 5,500 base repeats, a 2,000 base read isn't going to help us. Right? We only want the 6,000 base reads, because those will tell us the exact structure of the repeat with enough unique spanning sequence on either side so that we can unambiguously place it in the assembly graph, like Adam showed in one of the early slides. And what we realized is with the depth of sequencing they had for this genome, we had approximately 20-fold coverage in reads of 6K or greater. And given what Adam said before, that 10x of coverage should be good enough to get good assemblies, this 20x of 6KB reads should be good enough to get a very good assembly of this genome. And actually, in the future, we corrected the full data set and just took the long tail. You could just not even bother correcting any sequences that are less than 4,000 bases or even 5,000 bases if your goal is to do this kind of closure. So the result of our assembly was a single chromosome in one contig. So this figure just shows the annotated contig that we produced from the assembly. And again, I'll emphasize that this was a push-button automated process. So we ran the correction. We took the longest 6,000 base plus reads, and we ran the Celera assembler, and we got an automated result out. So this just shows some of the annotated repeats along the assembled contig. And what I wanted to focus on is some of these larger repeat structures. So you can see that we're able to correctly reconstruct these repeats that are typically very difficult for assemblers to reconstruct because they're 5,500 bases. So there's a lot of ambiguity, and there's a lot of places in the genome they can go. So you can see that the RNA operon, even in the small chunk of the genome, has 4 different copies. And typically, you wouldn't know which copy goes where in the genome if you have short read data. And in our case, since we have the 6 KB reads, we can unambiguously place them and reconstruct it and accurately get all the RNA operons placed along the genome. So this led us to kind of explore other genomes and see, well, is this a fluke? Can we do this in an automated fashion? So we developed a recipe for finished assemblies of simple bacterial genomes. What I mean by simple is ones where the largest repeat is approximately the RNA operon, or about 5,500 bases. This is a limitation of the PacBio sequencing. If you had a 10,000 base repeat, for example, you just wouldn't be able to get enough PacBio coverage that's longer than 10,000 base pairs with the current chemistry in order to unambiguously place that repeat. As the PacBio read lengths improve, this will automatically scale up. So the recipe doesn't need to change as the technology improves. You can keep using the same recipe, and the genomes that are simple will keep expanding, and the definition of simple will keep increasing and increasing. The other benefit of this is that this is closure using only PacBio RS sequencing without requiring any paired end or mate pair sequencing. So typically constructing, for example, an Illumina jumping library is significantly more costly and time-consuming than constructing a PacBio library or than an unpaired, for example, 454 library. So this is a way for us to save money and to speed up the processing of the assemblies, and that's one of our goals, time, not just cost. And our goal is to basically sequence enough PacBio to get 20-fold coverage of long sequences and span all the repeats. So we want to get 20-fold coverage of approximately 6,000 base pair plus sequences. Currently, this is approximately 200x, or about 8 to 10 cells of the PacBio instrument. Again, as I mentioned before, if the PacBio read length doubled tomorrow, you would only need to have 100x if it maintains the same distribution to get to this closed recipe. So again, it automatically scales up as the PacBio technology improves and the read lengths keep improving. You get automatic benefit without having to do new development. So this shows that we've applied this recipe to 3 different genomes with different combinations of data. So the first one is the coli K12, where we were able to close it by using a combination of PacBio sequencing with CCS data. Since then, we've also used 454 and MiSeq data to close the same genome and get the same 1 chromosome, 1 contig assembly. We were also able to close a Salmonella genome with a combination of 454 and the PacBio data. And I'll point out here that, as Adam mentioned, for these longer technologies like 454 and CCS, you don't need 100x of coverage. You can see that in 2 out of the 3 genomes, we have 30, 35-fold coverage, and we're able to close the genome. So you only need to go up to the 100-fold coverage if you have the shorter MiSeq data or other Illumina data when you're trying to close these genomes. And the last genome is the one that I started the presentation with. The first one we were able to automatically close in this way, and we used a combination of CCS or 454 and long PacBio data. And for comparison, I included what the best assembly we could get was with the complementary data alone, so the data we used for correction. And you can see in all cases, it's significantly worse. There's, in the best case, there's 25 contigs, so that means that's how many gaps. Basically, you have to look at all those gaps, figure out what goes in that sequence, and try to do manual PCR and other manual finishing processes to try to put that together into the one piece that we can get automatically. The limitations of this, obviously, is that currently we're targeting the largest repeat of rRNA operons, so this would not close the eukaryotic genomes, obviously, and it wouldn't work for more complex bacterial genomes with multiple phage insertions, segmental duplications, and other complex repeat structures. So we're looking into optical maps and other options to try to get those to the same kind of quality and finished state that we can get these simple bacterial genomes to. So the other thing we looked at besides these closed bacterial genomes was what we can do for eukaryotic assembly. As I mentioned, obviously, eukaryotic genomes are too complicated to have larger repeats, more complex repeat structure that we can't close their genomes, at least not for a while. What we did do, though, is we looked at the parrot eukaryotic genome assembly. And this project goal was actually, the goal of the project was to look at the vocal learning. And it's hypothesized that the vocal learning is very dependent on regulation, not actually on the exon sequence. So our collaborators were very interested in getting not only the exons in the genome, but also the intronic sequence and promoter regions of the genome, which are typically very difficult for assemblers to assemble. So what we have here is several assemblies that we performed. So we had a relatively low coverage of PacBio, less than 4-fold coverage. And we tried it in combination, correcting it both by 454 data and by Illumina data. And you can see that the 2 assemblies on the bottom, the one in green and this one here, these are the 2 that include PacBio data. This one is, the green one is corrected by Illumina data. The blue one is corrected by 454 data. And they're both significant improvements over the best assemblies you could get with just 454 data and just Illumina data. And they're actually better than previous bird sequencing projects which have been done with even Sanger sequencing. To put it another way, the assembly of the 454 data alone versus just adding 4x of PacBio closed over 50% of the gaps in the scaffolds in this assembly. So from just 4-fold coverage of PacBio data, you close half your gaps. And we wanted to see, well, what are the gaps we're closing? Are these gaps interesting? Is there any kind of interesting sequence that we're filling in that was missing before? And what did we actually fix? So we looked at the gene content using the genes from the zebrafinch project. And the figure on the right here shows the content of the exons within the scaffolds. So the takeaway here from the figure on the right is that Illumina is actually doing very well. It's the best one. It's the green line that's the highest here in this figure. So if you're after your exons, you can get them very accurately reconstructed in your scaffolds from deep Illumina coverage sequencing. However, on the left, we see that the Illumina assembly drops to the bottom. So here we're looking at contigs. So this means where you're able to assemble the exons and the introns between those exons accurately, and not just fill it in with ends because you didn't know what the intron looks like. And when we look at this metric, you can see that the PacBio assemblies, the assemblies that include PacBio, the red and the black lines here, are significantly above either using 454 data alone or Illumina data alone. So if you want to know not just the exonic sequence, like we did in this project, but the introns and regulatory sequences that surround them, PacBio data is very powerful and helps to fill in a lot of those intronic sequences that are otherwise missing from your assembly. And one great example of this is the FOXP2 gene. So this is an extreme example of a difficult-to-assemble gene. It's annotated along the bottom here. So these large vertical lines, these are the exons. The gene actually is about 400,000 bases in the genome with the expected short exons in the middle. So you can see that the vast, vast majority of this is intronic sequence between short islands of exonic sequence. And what we did is we looked at this FOXP2 genome. We first looked at it in the assembly of the Sanger assembly of zebrafinch. So this is the orange fragment here. And then we looked at the 4 assemblies we generated. So I'll go through and explain this figure in more detail. Basically, each one of these long blocks is a contig in the assembly. Whenever there's a break, it's a break in your assembly. So you can see that the Sanger assembly is relatively decent. There are not too many pieces here, but it can't reconstruct the full intronic sequence of this gene in one piece. It has breaks in some regions. Some of those breaks, for example, the break here corresponds to a higher GC window. So this track down here shows GC content along this gene, and there are higher GC windows in intronic sequence and maybe promoters regulatory sequence than in the exonic sequence. And you can see that the Sanger data has some trouble assembling in a particular high GC window here, as well as at the end here where it gets more fragmented as the GC bias increases. When we look at the Illumina assembly, we see that it's more fragmented than the Sanger assembly, which is expected. The other thing we see is that there are several gaps where we just didn't have, either the contigs were too short to be accurately mapped to the gene, or we didn't have any contigs in our assembly. So you're not only fragmenting it, you're missing some intronic information. So this is very difficult if you're doing analysis and you're interested in the introns. You're maybe missing parts of the story, and it's very hard to know that you're missing these parts if you didn't have a good reference or a good close neighbor. The 454 data helps improve over this Illumina assembly and fills in some of these gaps, but it's still relatively fragmented, and it still has issues in some of the high GC regions, the one I went back to. So this vertical line here, you can see that all the assemblies except one have a gap here because of that GC bias. The 2 by far best assemblies are the ones that include PacBio. The first one here is the 454 corrected PacBio, and this one is the Illumina corrected PacBio. So the Illumina corrected PacBio assembles this entire 400,000 base gene into one contig. So for the first time, even compared to Sanger data, for the first time, we have the full intron structure of this gene that we haven't had before. So this makes studying these genes much easier because you can actually see the regulatory sequence, intronic sequence that you couldn't get any other way. By any of the other assemblies, we're just missing this information. And one takeaway here is that the Illumina data does seem to do a better job than the 454 data with these high GC windows, so the gaps that are left in the 454 corrected PacBio assembly is in the higher GC regions, which is likely because of the fact that we have lower depth of 454 sequencing, and we couldn't have enough data to correct the PacBio accurately in those regions with high GC, whereas with Illumina we had enough coverage that maybe we didn't have the full depth of coverage, but we had enough coverage that we could accurately call the consensus of the PacBio corrected reads. And like I said, this is an extreme example, but it's a great example of the kind of benefit you get from this long reads in eukaryotic genome assembly that you wouldn't get otherwise. So in conclusion, what we wanted to demonstrate is basically that the high error doesn't prohibit assembly. So what's been holding back PacBio is that there have been no tools that can deal with the high error rate that they produce. But what we've done is fix the high error rate, and now it makes all the tools that have existed before, all the tools that are applicable to Sanger data, applicable to PacBio. So the potential applications that we've gone through, we can close simple bacterial genomes. We can significantly improve complex bacterial genomes and eukaryotic genomes, especially the eukaryotic regulatory sequence. We're hopeful that these long reads have other applications in the mixed sample preparation, like haplotype phasing, that you should be able to have much longer range information, so you may be able to better separate diploid genomes. And one other application that we didn't cover today, but we cover in the paper, is transcriptome annotation. So since these PacBio reads are longer than the typical transcript length, you can actually get the entire transcript and the entire variant of the transcript in a single sequence. And so you don't have to do any kind of assembly to get the transcript, full transcript. The remaining challenge is, obviously, since PacBio is a relatively new technology, there's some growing costs. So it's relatively low throughput now, and it's relatively expensive because of that low throughput. There's some learning curve that you have to get used to running the machines. The machines they're very sensitive to how they're run, so there's a lot of variation in the quality of the data you get out of them, and it takes it takes some time from any new technology when it comes out until it becomes mature enough to be robust and reliable and reproducible. And then there's some work remaining for us in terms of improving the efficiency of our tools, and also in best leveraging the high coverage Illumina data and the PacBio data together at the same time. So with that, I want to acknowledge all our collaborators. First, Mike Schatz and Dick McCombie at Cold Spring Harbor. Mike did a lot of our analysis with us, and they also provided the yeast sequencing. J. Craig Venter Institute, who maintained the Celera assembler, which we used extensively. The JGI actually performed all the analysis in our paper that was the transcript analysis. Duke University led the sequencing of the parrot genome and the parrot genome project, so they provided all the data for that. They did a lot of the analysis for that as well. And the University of Maryland, who provided us with E. coli sequencing data from the German outbreak strains. Finally, the USDA, who provided us with the genomes that we've been able to close. So they've been very heavy users of our pipeline from the start, and they've been closing genomes on their own. I think they're up to 7 or 8 now. And they were the ones who provided us the data that we've been using to develop our recipe for closed bacterial genomes. And Pacific Biosciences, who've been very forthcoming. They've shared a lot of data, shared their experiences. So they've been very great collaborators and very open. And the Assemblathon, who made all the data for the parrot public and available to everyone to work with. So with that, I'll stop here, and I'll take any questions you have.
Serge and Adam, thank you both very much. Kevin Davis back on the line. That was just a tremendous dual presentation. And we have indeed been inundated with questions, so I'm going to dive straight in, and let's keep the answers nice and crisp so that we can try and squeeze as many of these questions in as possible in the next 10 or 15 minutes. If we are unable to get to your question, or indeed, if you would like to request a reprint of the Nature Biotechnology paper that was recently published, it's not open access, but Adam and Serge will gladly send those on request, and will I'm sure, delve into any questions that are beyond the scope of this webinar or that we simply don't have time to get to. You can see their email addresses and contact information on the screen right now. So, okay, back to the top. We've got some sort of more general background clarification questions, and then some more specific technical questions for people who are actually trying to put this approach into practice. So some different degrees of difficulty, but let's just go through them. Some basic questions, Adam, firstly about, just so we all know what we're talking about, what are you seeing as the typical PacBio error rate? Can you talk about the distribution of those errors? Are they random, or is there an error profile? And perhaps you can just quickly clarify what is the read length distribution that you're seeing on the PacBio sequencer.
Sure. I think, Sergey, you still have control of the ball, so flip to the error distribution slide that's at the end, if you don't mind.
Sure.
So this plot, this plot represents the positional error rate bias in a PacBio read. So this is showing from read position 0 to 4,000 the empirical error rate that we observed in the yeast data. And this was an early C1 preliminary chemistry, so this data is now more than a year old, but it's still representative. The error rate hovers around 15% on average, but the important key point here is that there's not an observable positional bias. So unlike Sanger, Illumina, 454, every read technology we've ever had gets worse as the read goes on. A PacBio read is just as good at the beginning as it is towards the end. You'll see a little flare in the distribution at the end. That's just a statistical sampling artifact because there's less and less reads that are 4,000 bases, so there's more variance in the averages there. But in general, over the whole length of the read, we see a consistent about 15% error rate from start to end. The important part about that being a general kind of unbiased error rate is that it's easier to eliminate the errors when the error is unbiased, regardless of how severe the error is. In terms of the read length distribution, it's an exponential decay function, essentially, because the read lengths depend on the survivability of the individual. Looks like Serge is searching for a figure here. There you go. So the read lengths depend on the survivability of the polymerases, and as they're exposed to the laser light, they gradually die. And that's a random process, and it results in this kind of exponential decay here that you see, where the vast majority of the reads are short, but as you go down the distribution, you get a tail of reads that's actually quite long. So this is showing a real read distribution for this Bibberstania genome, and you can see on the right hand, the table is showing starting with 250x coverage pre-correction, the depth of coverage in reads of a different length or greater. So in reads 2 kb or greater, 171x. In reads 6 kb or greater, 21x. So the key of this is if you sequence enough SMRT cells with the PacBio, you'll end up with a considerable depth in very, very long reads. So having 20x in reads that are all 6 kb or longer is fantastic.
Do you anticipate, I mentioned nanopore sequencing in the introduction, do you anticipate a year from now, if and when these are in full commercial release, that you could, in principle, be able to substitute long nanopore reads for PacBio?
Yes, our approach doesn't care what the input is, and the approach is general enough that it will work just fine for any type of high error long read. So if Oxford Nanopore is able to produce reads that, say, have 10% error, and they're of equivalent length to these PacBio reads, our approach will work just as well as it's working on these, probably better.
Okay. Again, a couple of questions just to clarify items that I think you went over in your presentation, but just to make sure everyone understands what the workflow is. 2 questions. What aligner is used for mapping the short reads to the PacBio reads? And is the error correction performed after or before assembling the genome?
So the algorithm used is a seed and extend strategy. It's not a standalone aligner like Bowtie or BWA. It's part of the Celera Assembler's overlapping module, which has been around since the days of Sanger. But it just uses a hash seed and extend method, which pretty much all of the typical short read aligners use these days. What was the second half of that one? Of that question?
The second half, is the error correction performed before or after assembling the genome?
Before. Yeah, the error correction is performed before, and then the corrected reads are output, and they can be used for anything, either assembly or alignment or whatever you'd like to use them for. So the error correction module is completely separated from the assembler.
And the choice of Celera Assembler, obviously, as I mentioned in your bios, you have some great familiarity with that program, but have you sort of empirically decided that that is the best program to use in this approach?
That choice was primarily geared because we had experience with it, and so it was easiest to get that up and running the quickest. As I mentioned when I was presenting my slides, we've done some experimentation with using additional assemblers on the corrected reads. So my example for there, I believe, was Mira, which performed quite well. on the corrected reads, but there really aren't a lot of overlap-based assemblers or De Bruijn assemblers now that can even input long reads. So for example, the 454 assembler Nubler, I think its maximum input size is 2,000 bases. So those constraints affected our choice as well, and Celera assembler was one of the few that could handle long reads out of the box because it was designed originally for Sanger reads.
Let me just jump to another assembly alignment question that came in. If we can use MUMA to map reads, would something like Minimo be useful for assembly? Have you done a comparison of Minimo versus Mira versus the Celera assembler and or others?
Yes, certainly. So Minimo is an implementation of the unitagging approach, which essentially is what Celera assembler is doing. However, the implementation of Minimus and these other AMOS packages is done in such a way that it's not efficient for really large whole genomes. So anytime we're doing a whole genome approach of, say, a bacterial-sized genome or larger, we would like to use a production assembler like Celera assembler. Minimo, Minimus would work just fine for viral genomes or small bacteria, but really it's a scalability issue there. But yes, assembly of these corrected reads would work just fine in Minimus or Minimo.
Can you combine the HiSeq and PacBio for identification of alternative splicing and or fusion genes?
Yes. Serge, can you flip to the RNA Seek slide?
Yeah.
So in the paper you can look, we have a small section on RNA Seek, and here's the figure from the paper showing on the top, these are BLAT alignments of the uncorrected PacBio reads, and you can see that there's a number of exons that were not aligned properly by BLAT. In green, it's showing after correction how the transcripts align. So these are corn transcripts that were assembled by PacBio, sequenced with PacBio and Illumina. And in green, after correction, you can see all the exons are represented, and at the very bottom, there's two splicing variants where that next to last exon on the right-hand side is in a variant splice form. And the reference annotation is in gray on the bottom there, and you can see we picked up 2 out of the 3 splice variants in the post-corrected data. So we are starting to do more experiments with RNA Seek, but these are preliminary results. Fusion genes, fusion transcripts, cancer cell lines are all things that we're confident our program would work well on, but we haven't done any work with the actual data yet.
Is it implied that the user sequence his or her genome, the genome under investigation, on both PacBio and Illumina before assembly? Or by short reads, do you mean short reads that are produced by the PacBio instrument?
It can be either way. So we're agnostic both to the single molecule technology, so we could use PacBio or we could use a future technology like Nanopore. And on the short read side, as long as it's a high accuracy read, we can use any technology. So we've presented results in the manuscript both correcting with Illumina, correcting with 454, and correcting with the PacBio CCS. In fact, the results using the PacBio CCS were some of the best of the results. It worked quite well for the correction. So you could do it all on one machine.
Great. 3 questions I'm going to clump together. I think they all touch on the same issue about ATGC content and potential bias. One is, wouldn't you also potentially lose some areas where the PacBio was able to sequence and the Illumina was not, such as high ATGC? How sensitive is error correction to uneven coverage in the high accuracy reads? And Erwin wanted to know, how do you deal with regions in the genome that are underrepresented in the short reads, e.g. high or low GC regions, or regions that can't be amplified well due to secondary structures? Can you kind of talk about that?
Yes, this was a weakness of our original approach, in that if there was a bias or there was missing sequence in the secondary, say, Illumina data, that would be kind of projected onto the PacBio reads, and you would lose some sequence in those areas. Recently, Sergey has added a module to the pipeline that in areas where there's no coverage of secondary sequencing, we can use the PacBio reads themselves to fill the gap. So Serge, can you slip to the consensus figure? So even though the individual reads have a high error rate, as the coverage of even the high error PacBio reads increases to above like 3 or 4x coverage, you can start to knock out the errors in the PacBio reads using only the PacBio reads themselves. So we've added some recent code to the pipeline that in areas where there's coverage gaps in Illumina, we can use the PacBio reads themselves to form a consensus sequence, and that's been working quite well. In cases where the coverage is biased but it's low, but not zero, the pipeline works fine on that. It doesn't seem to be terribly affected by what the coverage variations in the Illumina or the 454 data look like. So if you have lower coverage in some regions, that'll be projected onto the PacBio reads, and the result of that region will be depth in whatever the PacBio depth was originally.
Adam, in your presentation, you mentioned there was a recent paper from investigators at the Broad on using all paths in a complementary approach. There was also a paper authored, I think, exclusively from investigators at PacBio in the same issue of Nature Biotechnology. Are there pros and cons to all these various approaches, or would you, I know you're biased, but would you suggest that your approach is more, overall, has advantages over the other two?
Yeah, no, I think there's pros and cons for both of the methods. So the other method that was co-published with ours was Allura, and that's based on a lot of the AMOS tool packages that Sergey and I had helped develop. So we were kind of tangentially involved with that project as well. And the all-paths LG approach, I'm not biased in saying that I find the all-paths LG assembler to be very, very competent. And we had a gauge competition. There's a paper in Genome Research with Steven Salzberg, myself, Sergey, and others, where we compared short-read assemblers and came down on the side of all paths as being kind of the most competent Illumina assembler around these days. So I'm very high on all paths. The cons of doing the approach of assembling first and then trying to scaffold with the PacBio reads is that I think it's going to be hard for that to scale. So for instance, the Allura assembler and the all-paths LG PacBio assembler both are limited to bacterial-sized genomes. I believe this is kind of a fundamental limitation because the problem of mapping long reads to a De Bruijn graph is a hard one, and I think it'll be hard to do that for eukaryotic-sized genomes like the bird. So for large genomes and for cases where you need flexibility, that's the other issue. The all-paths assembler requires pair information. So if you just have unpaired Illumina or unpaired 454 or PacBio CCS without pairing information, our method is really the only option for those. If you do have kind of a production setting where you're able to make jumping libraries with Illumina and you're able to make the short jump libraries, and you can combine all of those data types, I would not hesitate to recommend all paths. I think it's very competent. But there's definitely drawbacks to each of the approaches.
Yeah, the only thing I would add to that is that if you look at the all paths approach, they actually rely on the Illumina jumping libraries to resolve the repeats and the PacBio reads to fill the internal structure of the repeats. So I think this gets back to the point that Adam brought up, that as these PacBio reads get longer and longer, it becomes more difficult to figure out what path they correspond to in the De Bruijn graph because the path is longer and longer and there's more and more options to go through to get from one end of the read to the other. So I think that's a limitation. I think that's one of the main differences between our two approaches, that we can automatically scale sort of agnostically with the read length, whereas there it becomes more computationally expensive. And I'm sure as PacBio reads grow longer, they'll adapt their assembler to work with that as well. But at the current, at least the state of the art, the way it is now, they rely on shorter PacBio reads than we do.
Okay, great, thanks. We've reached the top of the hour, but we're going to go into overtime because we still have a bunch of questions. So I hope our presenters and everyone else can just hold on. We'll have another 5-10 minutes at the most. We'll wade through the other questions we've had. I've got one.
May I nominate a question quick, Kevin? That,
you may, quickly, yes.
Anne had posed to me that I'd like to address. She had asked, she has a single-cell sequencing project. She has some DNA. She's wondering, due to the bias of multiple displacement amplification, would you recommend using HiSeq or PacBio CCS for the sequencing? Sergey can talk a little bit about single-cell. He's been working on that recently. The point being here is that the bias is removed in the sequencing process. So any of the bias that's introduced by the MDA itself will be replicated on whatever platform you use. So if you have a single cell and you need to use MDA, there's really no way to get around that original bias. So sequencing the resulting product of the MDA can be done on either or any platform. The PacBio reads would give you the advantage of getting a more complete assembly of whatever single cell you're doing. Sergey, do you have any comments on single cell assembly while we're talking about it?
Yes, I've been looking at some single cell assemblies from data generated by the Bigelow Laboratory in Maine. And the basic takeaway is that, well, one issue is that, at least in their hands, the PacBio required more DNA going in, so they had to do more amplification, so that did increase the bias in the PacBio data versus the Illumina data. So that's a matter of how much DNA do you need to go into each instrument. If you need more DNA for one instrument than another, you may need to do another round of amplification, which will add more sequencing bias by that process. The other kind of main takeaway is it's sort of a question of what are you after. The same as in the eukaryotic genome, we saw if you're after the exons, the Illumina data at high depth works great. And it's the same thing here in the single cell. If you're after the general genes of the genome but don't care too much about the full structure or the repeats within it, the high-depth Illumina sequencing will beat the PacBio because you just can't get enough depth of the PacBio sequencing to get around the MDA bias that you have. However, in the assemblies I've looked at, none of the Illumina data can assemble through the larger repeats, and when I say larger, they're in the 2K, 3K, 3,000 base range, whereas all the PacBio assemblies can get through these. So it's really a complementary technology, and it's a question of what you're after. If you're after just the gene sequence, don't care too much about the full structure and the repeats, the Illumina data at high coverage is your best option. If you're interested in the repeats, you can add some PacBio sequencing to that to get through the repeats and combine it with the Illumina data to get the best of both worlds.
I'm going to ask a very naive question, which is we've been talking, I think, exclusively until this point about de novo genome assembly. Does your approach offer any advantages for example, human resequencing, where you've got a reference genome?
So it depends, again, what Serge just said, what you're looking for. If you're looking for SNPs, human resequencing, I think far and away is the better option is to just do Illumina or complete genomics or solid or whatever your short read preference is, because those do a very good job of pulling out the exonic SNPs and things that you care about. With the reference genome, it's probably not necessary to use a long read technology because you already have the structural information. If you're interested in things like haplotype phasing or structural differences versus the reference genome, then yes, the PacBio reads would give you an advantage there if you were looking for any kind of variable number tandem repeat profile changes, any kind of chromosomal disorders, fusion genes, anything that has a structural component, you get an advantage with the long reads. But if it's just a population SNP study, then short reads are the way to go.
Thanks. Matthew was asking about computational requirements, memory, and so on. He was saying that a typical experiment was going to take about 2 months on a 32-core machine. Can you talk about what compute resources may be required to really take advantage of this approach?
Sure. Bacterial genome takes about an hour, so no more than typical assembler. I think that's what All Paths was reporting for its PacBio bacterial assemblies. So bacterial assemblies can be done on a single compute server. If you're trying to do a gigabase genome like the commenter was attempting, like the parrot genome that we did, that required us to run on hundreds of cores for about a week. So if you're doing a large genome project like that, you should really try to secure access to a compute cluster to do the correction phase. We're working on speeding up as much as possible the aligning and the correcting of the PacBio reads, but for those large genomes, you do require some server size. I'll say also that just for assembling genomes of that size requires significant compute power. So assembling the PacBio data from 454, just using Celera assembler, took a week on a cluster. So if you're in that large genome space, you kind of need the compute hardware to back it up. Do you have any comments on the runtime, Serge?
Yeah, so the thing that I wanted to add is that Celera assembler has an unfortunate learning curve in that you can very easily configure it wrong and make a data set that takes an hour in someone's hands run for a week or two in someone else's hands, because it's very easy to over or underload a machine and significantly impact your performance. So we're aware of that problem, and we've been working in collaboration with PacBio to try to automate some of these decisions so that the person doesn't have to tune it just right. They can just give some general information about their machine in terms of cores and memory, and the pipeline will automatically tune itself to maximize those resources. So I think that's, in the support issues that I've had, I think that's been one of the biggest issues, and unfortunately Celera assembler has had this learning curve for the last 10 years since it's been written. So that's been the biggest, I think, pain in terms of performance.
Yeah, and certainly we encourage you to just send us an email if you have any trouble tuning the pipeline. We're always happy to help our users.
Have you tried using the cloud for this at all? Any reason you couldn't?
I think PacBio themselves are looking at that. I think they are looking to have their analysis toolkit available on the cloud. And also there is a virtualization project called Clover, C-L-O-V-R, that was written by friends of ours at the Institute for Genome Sciences at University of Maryland, Florian Frick and Sam Angioli and company. They have Celera Assembler included in their virtual machine distribution, and in future versions, I believe they're planning to include our correction pipeline as well. So in that case, you would have a virtual machine that you could fire up on the Amazon EC2 or any of the cloud-based platforms and fire off a correction there. So that should be a good solution for people that don't have institutional resources available.
All right, just a few questions now in closing. We'll just keep the answers nice and short, and then we'll wrap up. And obviously, if we don't get to your question, please feel free to email Adam or Serge offline. Bob asks, if you clip parts of the PacBio data that don't have Illumina mapped reads, do you see issues where the reason for the absence of the Illumina reads is ACGC content, so it is real sequence? Grammatically, I'm not sure if that makes sense, but did you get that?
I can speak to it a little bit. So there was regions. Okay. I think the question is when there's gaps in the Illumina coverage, what's causing the gap? Is it a GC bias problem? And in the FOXP2 example that Serge showed and some of the other regulatory regions of the parent genome, we did see that there was very high or very low GC regions that caused gaps in the correction. And we were only able to fill in those regions by having high-depth Illumina sequence. So Illumina actually came in helpful in some of those cases. You can see there's a spike down here at the bottom in the GC plot that corresponds to some of the gaps. So definitely GC content can affect the coverage of the short read technology and lead to gaps. But as I mentioned earlier, Sergey has recently implemented a part of the pipeline that can close some of those gaps using the PacBio reads themselves.
If you have 10,000 PacBio reads, how many reads will pass through your error correction algorithm?
So this is what we've been calling throughput, the number of reads that are successfully corrected by the pipeline, and it entirely depends on the quality of your PacBio run. So we've seen throughputs as high as 80 or 90% of the reads being corrected, and we've seen throughputs as low as 10 to 20% of the reads being corrected. So it entirely varies based on the quality of the sequencing run. If the reads are short, they get discarded by the pipeline, so we don't even bother correcting things that are less than 500 bases, just because there's not a lot of use in correcting things that are shorter than your Illumina or 454 data, for instance. So on average, probably about 60 or 70% of the data you'll be seeing get corrected if the pipeline is operating normally. If you see much less than that, take a look at the quality of your PacBio run or contact us. You might have some configuration problems with the pipeline.
And I think we'll have to make this the last question as we run significantly over time. Have you compared your method to the new p-error correction module from PacBio?
No, I don't think we have. I know that they did an error correction step when they did the E. coli German outbreak study last summer. And that consisted of an approach very similar to ours. And in fact, it was inspired originally by a presentation of ours we had done in May of 2011. Where we had been collaborating with their assembly team on some methods. I believe they did a similar approach where they take the short reads and they align them to the PacBio reads using their Blazor aligner. Then they call a layout similar to how we do. And then I believe they assemble using some of the Amos assembly toolkits. So we haven't directly compared, but I suspect it would produce similar results, but the assembly process itself might be considerably slower than ours.
Okay, I think let's call it a wrap there. Adam Philippi and Sergey Koren from NBACC, that's just been a tremendously useful presentation. We had over 100 people on the webinar today, so I hope everyone found it constructive. If we weren't able to get to your question, please feel free to email Adam or Sergey. We'll put their email address up if we can. Just one of the
Sergey, can you flip it to the slide?
Sure.
So if you would like to ask them a question offline, Adam and Sergey will gladly answer questions over their lunch break, and be happy to send you a copy of the Nature Biotechnology paper. Let's say one of the landmark papers in next-gen sequencing this year so far. On the chat dialog box on your screen, there is a link to a survey. We'd be very grateful if you could take a minute or two to follow up with us, send us your comments on today's webinar, and send us suggestions for other topics that you might like to see covered by NGS Leaders. They are graciously making this webinar platform available for breaking news and high-profile studies, and we'd like to continue that trend as we feel this is a very valuable use of the community, and we hope you enjoyed taking advantage of it. A final reminder that next week in Providence, Rhode Island, it's the CHI Next Gen Sequencing Conference, a fantastic 3-day conference. The opening session will include presentations from leaders at all of the major genome centers, including the executive director of the New York Genome Center. So if you fancy learning about and possibly even applying to one of the high-profile positions at the New York Genome Center, I think it might be worth a while popping along. We have a discount code, NGS200. NGS200 is the key code to enter for a $200 discount if you want to be a last-minute registrant at that conference. That does it for me, Kevin Davis. I want to thank Jean Holly and my colleagues at NGS Leaders, and once again, Sergey Koren and Adam Philippi for a fantastic presentation. And thank you, most of all, to you for hanging in there and joining us today on this NGS Leaders webinar. We hope to bring another one for you in the very near future. So for now, thank you for joining us. Goodbye. Thank you, Adam. Thank you, Sergey. All the best.
Thanks, everyone.
All right. Bye.